Okay, hi everybody. Today we're going to talk about cellular respiration, and this is one of the most difficult topics that we've covered so far. So what happens starting right about now in my general bio course, we hit cell respiration, and then from there on out we hit several of these biochemical pathway topics, and they tend to be quite difficult. So we're going to give it a shot, and we're going to cover it the best we can and see what we can get from it. So this topic again is called cellular respiration, and this is the equation here. And basically what we're going to do is we're going to take some of the food that we eat and we're going to break it down to exchange the energy in the food we eat, and we're going to convert that energy into another type of chemical energy that we call ATP. So that's our goal. That's what we're attempting to do there. Okay, so you need food for all the various things that you do. Obviously, you need it for the building blocks, for the chemicals, but you also need it for the energy. And that's what we're going to focus on today, is the energy aspect of it. So a bird might eat food because it needs it for reproduction. A dog might jump in the pool after its favorite ball. And you might wear an elf onesie and learn how to play trumpet. And all those things are going to require energy, and there are some byproducts, things that come out of that. We're going to take oxygen in the case of this animal here, and we're going to release carbon dioxide and water as we go. Now, first, since we're talking about making ATP, let's talk about what that looks like and what our goal is for that. So we have two molecules. One's called ADP, one's called ATP, adenosine diphosphate and adenosine triphosphate, and they're basically the same molecule if you look, but the difference is right here, this third phosphate group and ATP, and it takes a lot of energy. It's an endergonic reaction to put that third phosphate on, and so because you put so much energy into that, there's a massive amount of chemical energy stored up and how they share that bond, which we'll talk about in a minute. So ADP, which is sort of an energy poor version of the molecule, and ATP is the energy rich version of the molecule. What we're trying to do is take the energy from the food we eat, and we're trying to convert that into creating ATP, or converting our energy from the food we eat into ATP from ADP. So that's what we're after there. Now, we talked about glucose. That's what we're going to start with. You can do this with really any kind of food molecule. So you can do this with proteins and fats and other things. Glucose sort of takes you through all the steps. So teaching wise, that's how we cover it. But nutrition wise, when you're eating, you can actually use other molecules that you eat and convert them into either intermediates into the steps, or you can convert those into glucose. There's a variety of ways it can happen. So this is glucose here. This is what we're going to start with. And when we talk about energy being stored in it, I'd like to point out what we're talking about is in the covalent bond between molecules, the way they share the covalent bond is where the energy is at. So the energy is, again, the ability to do work or bring about change in opposing force. And that energy is, in the case of chemical energy here that we're talking about here, it's actually already stored up in how they're sharing those electrons. So when we break that bond, that's going to release the energy, and we're going to try to capture that with other chemicals. That's our goal. Okay, so here's a breakdown of all of cellular respiration, showing you the basic parts. A lot of people and a lot of books will teach this as a 3-step process with a transition step or an intermediate step. They call it different things. I go ahead and just say that there are 4 steps to make it easy. So I'll label them 1, 2, 3, and 4, and then we'll go over each of them. They are glycolysis, the pyruvate dehydrogenase complex. They are glycolysis, the pyruvate dehydrogenase complex, citric acid cycle or Krebs cycle, and then the electron transport chain. And you can see down here how much ATP we get out of each of those steps. It's the last one where we're getting the bulk of our ATP. So that's worth noting there. So let's dive into glycolysis and see what we're talking about here. So we start off with glucose, and then we use the enzyme hexokinase, which uses up a little bit of ATP and generates it into ADP, and that gives us glucose 6 phosphate. And then what we do is we use another enzyme, phosphoglucose isomerase, which converts it into fructose 6-phosphate, and then phosphofructose kinase 1, which gives us fructose 1,6-biphosphate, and then aldolase and triose phosphate isomerase, which then gives us dihydroxyl acetone phosphate, which can then be converted into glyceride 3-phosphate. 2 molecules we split glucose in half. And that's what I'm talking about there as it gets complex. So if you take majors biology, you typically learn maybe all the steps of glycolysis. If you take biochemistry, you're learning all the steps and then probably all the enzymes that go along with it and how much energy and whether they're reversible and things of that sort. So it gets overwhelming very quick, but let's keep going and then I'll break it down. I'll tell you what we're going to do with it. So then our intermediate at that point, we have our glyceraldehyde 3-phosphate. Then we use our enzyme glyceraldehyde 3-phosphate dehydrogenase, which then allows us to take NAD and convert that into NADH. Now, why is that important? Well, that if we zoom in and look at that, what is NAD and what is NADH? If you look, it's kind of like ADP and ATP. NAD is an energy poor molecule, and NADH is that same molecule, and we've transferred the high energy electrons to it along with our hydrogen atoms that we attach to it. So really what we're doing is we're converting that energy that is stored up in our glucose, and we're using that to make NADH, which comes up later on at the end. So you continue all the way on to the end, you see here, and you get down to the end to having 2 molecules of pyruvate. Now, what I like to do, because like I said, it's very overwhelming when you look at all the pieces all together like that, what I like to do is I like to go through all the steps and show you all the pieces of it. But for my general biology class, for my non-majors class, we'll walk you through all the steps and show you them, but then I have you just learn, okay, for each step, we start with glycolysis, what goes in, what comes out, where does it occur, and what's our main goal of that. So what we're going to do is we're going to take glucose, that's what's coming in, and whenever we have NAD and ADP, we'll see that those kind of go along together. But what comes out of glycolysis is pyruvate. That's what's at the end here that we talked about. That's the last step of glycolysis. But we're making NADH and we're making ATP, and it occurs inside the cytoplasm of the cell. And the most important thing is, our goal is to make ATP, so that's important. But we'll see in the long run, it's this NADH that ends up being very significant, that we need that for that last step. So rather than learning all the pieces and all the steps, focus on what goes in, what goes out, where does it occur, and what was the main goal of that set of reactions. And then it's much more manageable than trying to learn all the intermediate steps and all the enzymes that go along with it. So focus on that chart as we go. Next is what we call the PDC, or the pyruvate dehydrogenase complex. Many books, again, don't even mention this. Instead, they'll call it an intermediate step or a transition step. And so I just label it here. This is what it looks like when you look at the pieces of it and the enzymes that are involved. But once again, what we're going to try to do instead is focus on, well, what happens? What's our goal and what do we do there? So if we ended with pyruvate before, or pyruvic acid and glycolysis, same kind of thing, then in the PDC, we're going to take the input's going to be our pyruvate, and out of that is going to be a 2-carbon molecule called acetyl coenzyme A. So we're going to break off one of the carbons, and that is where, in fact, we give off some of the CO2, some of the carbon dioxide comes off, because we take a 3-carbon molecule, we take 2 of them. Remember, we had glucose and we split that into 2, and so we had 2 3-carbon molecules from the 6, and now we have 2 carbon molecules because we're giving off CO2 in each one of those pyruvates. So we make acetyl coenzyme A, we make some NADH again, and this occurs inside the mitochondria this time. And the goal here, once again, we're trying to make NADH. The other part of the PDC is we're trying to transport that into other parts of the cell so that we can go on with the next step, which is the Krebs cycle. Okay, so citric acid cycle or Krebs cycle. And once again, you noticed we ended with acetyl coenzyme A, so we're going to start there. First thing we're going to do is transfer that to an intermediate molecule, oxaloacetate here, and we're going to make citrate out of that. And then that's going to be our carbon intermediate molecule there, our 6-carbon intermediate molecule there, 1, 2, 3, 4, 5, 6. Okay, so we're going to make that citrate, and then again there's a cascade of enzymes that convert this from one to another: isocitrate, alpha-ketogluteric acid, succinyl-CoA, coenzyme A, succinate, fumarate, all the way through. And once again, as this happens, for each one of these steps there's an enzyme and an intermediate but again, if you're a bio major, probably need to learn all the intermediate steps, and if you're taking biochemistry, you probably most likely will learn not only all of the intermediates, but also all of the enzymes that go along with it, and different parts of the reaction that occur. But for non-majors bio, once again, I like to take that chart and instead focus on, okay, what ended up happening, what did we do, and what's our chart look like. So in the Krebs cycle, we're taking in, once again, since we ended with acetyl coenzyme A, that's the first thing coming into the Krebs cycle or citric acid cycle is acetyl coenzyme A, and then we're going to take NAD, and we're going to make NADH out of that. We've done that before. And then you'll notice that we have this FAD and ADP. So we talked about that before, but what is the FAD? Well, if we zoom in, you can see that our FAD that we're producing over here, our FAD is very much like NAD. It's actually very, very similar, but it doesn't quite have the energy content level of NADH. So FAD becoming FADH2 is once again, it's one of those energy storage molecules. We're transferring the energy to the FAD to make FADH2. So it's very much like NADH, but it doesn't quite have the same sort of push, as you'll see in a second. And we're going to make, in the Krebs cycle, we're going to make 2 ATP. We're going to make, we're going to give off some carbon dioxide right there. And our main goal is, once again, in the Krebs cycle, is to make ADP, to make NADH, and also I might throw in this FADH2. And this is going to occur, once again, inside the mitochondria like the PDC did. Now, if we take a closer look at the mitochondria, we'll see that it's actually quite different in different parts of it. So the mitochondria actually has 2 membrane layers to it. So it has an outer membrane, and then it has an inner membrane that has all these folds that occur in it. And the folds we call cristae. And in the middle, in the very, very center, inside both membranes, is the matrix. So when we talk about inside the mitochondria so far, we've been talking about things that are inside that matrix. But what we're going to do on our last step is this process is going to take place along that inner membrane. So if I go back for a second, the last step is occurring along the parts of this inner membrane. That's where it's going to take place. Okay, so here's my inner membrane right here, and what's going to happen in the final step in the electron transport chain, what we're going to do is we're going to take our NADH and our FADH2 that we've been building, okay, and we're going to transfer the electrons. And when we do that, that energy, as we transfer the electrons, we're going to use that to activate these proteins that are going to be a type of protein transport pump. They're going to pump hydrogen ions across that membrane, from the outer membrane, from the inside, from the matrix, across the inner membrane into the space between the 2 membranes. And that's what NADH and FADH2 do. And so as the electron is transferred down from protein to protein to protein, there's a series of proteins in this step. It goes down and it continues to pump hydrogen ions across that membrane. And what ends up happening, we'll get to that in a second. So we're building this big surplus, really, of all these hydrogen ions. We're putting a almost like we often will describe it as sort of a waterfall, but it's not really water, it's hydrogen ions, and it's not up in the air, but instead it's a chemical gradient is what we're building of hydrogen ions as we're pumping them across there. And then at the very, very end, the very last step of that protein, the very last thing that comes along and grabs that electron as we've passed it along is oxygen. So oxygen comes in, grabs that, it's what we call the final electron acceptor. Oxygen grabs that along with two hydrogens and it forms water. So when you talk about needing oxygen, this is why you need oxygen. You need oxygen because it is that final electron acceptor in the electron transport chain, and that's what keeps the thing going and allowing us to build all these hydrogen ions. And without oxygen, this entire chain gets backed up and it shuts this down, which we'll talk about on another time. So now we have all our hydrogen ions built up, and what happens is there's a protein here called ATP synthase, and this protein does exactly what it sounds like it does. It synthesizes ATP. It's like a little motor, and it rotates. And so as the hydrogen ions, which we've built up this big supply of them, as they flow through here, it turns this sort of protein motor, and the energy in that allows me to take this third phosphate here and stick it onto my ADP to make ATP. And remember, that takes a lot of energy to do that, and that energy comes from those hydrogen ions flowing through, which ultimately that energy came from the bonds stored up in the food we ate, and we converted that into NADH and FADH2, and that end result is that's where you get most of your ATP. So in the electron transport chain, we have NADH and FADH2 coming in. Notice those were outputs before, those were important before, and so we're actually bringing those in now. Those are inputs, and so after we use up that energy, we're giving off NAD and FAD, and we're going to bring in oxygen, and we're also going to use this ATP synthase protein that's there. That's an important part, so I added it there. And what we're going to do is the output is we're going to generate ATP, and don't forget an important output is water, because that's formed as that final step. And we're going to then recycle and have NAD and FAD come out as outputs before. And again, this is going to take place along the inner mitochondrial membrane. So there's two membranes, and it starts in the matrix and it takes place across that inner membrane. And what's important here is this is where we make most of our ATP. So in a typical molecule of glucose, we might get 2 ATP from the glycolysis, and we might get 2 ATP from Krebs cycle, and we're going to get 32 to 34 or so ATP. Kind of depends. People kind of argue about exactly where that's at. We get 32 to 34 ATP from the electron transport chain. So I hope that was helpful, and good luck. And next time we tackle photosynthesis, have a great day.